All right, we will go ahead and get started. Welcome to the LRIG Philadelphia Science webinar. My name is Lorena Kalau, and I am at GlaxoSmithKline in Collegeville, Pennsylvania. I am your WebEx host today. Just a couple of things about the WebEx. First, we are recording the WebEx for future viewing. Secondly, participants are muted, so for questions at the end, we will be using the chat function. Please select to all panelists from the drop-down menu, and one of the panelists will read the questions to the speaker. With that, I would like to hand things over to the chair of our LRIG Philadelphia chapter. Over to you, Rodney.
Thank you. Good afternoon, and welcome to LRIG's fifth virtual scientific meeting. Today, this scientific meetings are basically an opportunity for scientists to share their research and innovations with the global LRIG community. If you are interested in presenting in a future virtual scientific meeting or organizing a future scientific meeting, please just email your proposal to talks.phl@lrig.org. You can learn more about our future meetings by going to lrig.org, and there will be a posting of any future events, which will have links to where you can register. Today, our meeting is co-sponsored by the Pennsylvania Drug Discovery Institute, and we are fortunate to have Dennis Gross, who's the CEO of the PA Drug Discovery Institute, who will introduce our speaker for today. On to you, Dennis.
Okay, thank you, Rodney. As Rodney mentioned, I'm the CEO of the PDDI. We are a nonprofit organization based at the Pennsylvania Biotechnology Center up in Bucks County, and we're involved in helping startups, engaging with STEAM education, as it's now called, at local secondary schools and even at colleges and universities with workforce development programs. And also, I serve as the co-chair for the National Biotechnology Entrepreneur Bootcamp, which is run in conjunction with the International Bio Meetings. So our speaker this morning, Andrew Reaume, is someone I'm proud to call a colleague, a very good friend, someone I've traveled with a number of times, and a great example of a highly creative entrepreneur, which is really something to consider in this environment we're in. Andrew has his doctorate in genetics from the University of Connecticut, went on to get an MBA from the Wharton School, graduating with honors in entrepreneurial management. And after grad school, he went to Cephalon and worked in Cephalon as a bench scientist, but also became very involved in identifying and coordinating in-licensing opportunities, a very critical phase of research opportunity looking for pharmaceutical companies. After he was at Cephalon, he moved on to Pfizer, where he got involved very extensively in some of their business activities and did some very significant things at Pfizer. He is, as Rodney said, the CEO and president and co-founder of Melior Discovery, a local firm, which was established originally as a biopharmaceutical company dedicated to in vivo phenotypic screening. And now he's moved into a very interesting and very important area, which is drug repositioning, or some people call it drug repurposing, but repositioning is probably a much better term for this. So under his leadership, he's actually advanced a number of compounds from discovery stage R&D into 2 successful phase 2 studies that have been done in a number of countries worldwide. So without further ado, I'd like to provide the floor, proverbial floor, to Andrew to talk about this area in a very important and very necessary area of looking at some diseases that are either untreated or undertreated, and looking for existing compounds that need to be repositioned. And he has become a master of identifying these compounds and working in a very structured environment to bring them forward into the clinic. So without further ado, Andrew, the floor is yours.
Thank you, Dennis. Thank you for those very kind words. I'd like to begin by just making a simple but hopefully provocative statement. I'll try to make it provocative, and that is just simply that biology is complex. But more specifically, what I mean by that is that there is much more about the biological systems that we're studying than what we don't know than what we do know. And more specifically, what I'm referring to, just to dig into this concept a little bit more, if we think of sort of the archetypal drug versus disease process relationship where we have an archetypal drug interacting with an archetypal receptor or target and working through some biochemical pathway that impinges upon a disease process, where this figure depicts that relationship. And for example, there may be some relationship between the pathway that we are studying and some other pathway that impinges on another disease process that would lead us to develop hypotheses on how that drug might have alternative therapeutic application. All, as I say, depict by this sort of archetypal way that we think of things. What I'm proposing is that the real picture is better represented by something like this, wherein what I'm saying is there's much more that we don't know, many more systems and interactions that we're not aware of that can either invalidate the hypothesis or in some way provide complexity to the outcome than what we would predict. So another way of sort of stating that is it's a case of we are effectively looking for our proverbial keys under the proverbial street light. That is, we're looking in areas because that is what we know, or not because when in the case of the key, we're looking for them underneath the street light because that's where the light is, not necessarily where we lost the keys. And I think the same sort of analogy applies to some of our research. But the emphasis I want to make is that there's much more that we don't know than what we do know. So I'd like to just pause on that for a second and then talk about one other thing, and that is the concept of pleiotropy. This is a term for those who might not be familiar with. It comes from classical genetics. It's a term that's been around for well over 100 years, but it refers to the phenomenon of a single gene influencing two or more distinct traits. So for example, something like hair color and intelligence or something, things that are seemingly quite unrelated. What I'd like to introduce for the purpose of this talk is a concept of pharmacological pleiotropy, meaning the notion of a single drug having potentially multiple, even quite unrelated therapeutic benefits, which should not be sort of a far leap or conceptually a big leap if you consider that in the case of genetic pleiotropy, this is a phenomenon, which, by the way, is maybe more of a rule than an exception in the case of genetic expression. And so it follows, insofar as these genes and the products of those genes are proteins, it follows then that that those proteins that have a wide sort of biological effect, that the same phenomenon should carry forth when we're modulating those protein targets with drugs. So, but to bring those two together, those two concepts, the biological complexity and this term I'm introducing, pharmacological pleiotropy, what I'm suggesting is that there's much more potential benefit that we just as a field do not appreciate that can be sort of harnessed out or tapped into with the drugs that we're working with. And it's by virtue of the sort of diverse biology associated with many of the targets that we're studying that we don't as a field fully appreciate. And so what I'd like to do for the rest of my talk is provide illustration of that. And I'm going to do so using one target that I'm going to take you on a tour through of multiple stories that I think illustrate this well. So the target I'm going to talk about is one that I've and Melior Discovery has been working on for quite some time, LRIG Kinase. And I'd like to take you back to the very beginning in as far as when the field of science first became aware of LRIG Kinase. So it was first discovered back in the early 1980s. In a lab that was focused on hematopoietic cell lineages and some subsequent studies in those labs and others that were doing related work on cancer cells, looking at upregulation, downregulation. But the point I'm trying to emphasize is that in some ways this was a key, looking for keys underneath the streetlight scenario again, wherein because of sort of the way it was discovered and the interests of those researchers, the field remained quite focused for a period of on the order of about 20 years, just looking at Lin kinase function in that setting, which is to say that that's not necessarily reflective of where Lin's most important function may lie, but that's where the interest of those researchers was. And they described it as having an inhibitory role in myeloid cell proliferation. But in about 2000, an investigator by the name Mueller, not necessarily interested in Lin per se, but what he was interested in was studying a drug called glimepiride, which for those who aren't familiar, it's in the sulfonylurea class. It's a insulin secretagogue, meaning that it promotes pancreatic beta cells to secrete more insulin. But it was known that glimepiride had glycemic control activity that couldn't fully be explained just by its role in causing pancreatic beta cells to secrete more insulin. And that's a phenomenon he was sort of digging into. And to make very long story short, he discovered that there was an effect taking place in the insulin signaling pathway, and he narrowed it down to a single, or at least a key factor, which was Lin kinase. And what he showed is that glimepiride was having an indirect, non-specific activation of Lin by disrupting the lipid rafts that Lin reside on. And by disrupting those lipid rafts, it was causing this activation effect that in turn had a positive influence on insulin signaling. Independently, and some years later, about in around the 2005 timeframe, Melior Discovery became involved in Lin kinase research. So our story began with working with a drug we've called MLR1023. This was an old Pfizer drug that had been pursued as a gastric ulcer therapy up through phase 2 clinical trials, had a lot of favorable attributes in terms of being safe and well tolerated, but wasn't fully effective in gastric ulcer and was discontinued for that reason. But otherwise, as Dennis was sort of alluding to, we have an interest in drug repositioning. It fulfilled key criteria that really made an ideal repositioning candidate. It never had market exposure, but was clearly biologically active and well tolerated in the clinic. And so we picked it up and did what we are really pioneers in, which is in vivo phenotypic screening of reposition candidates. And so what I'm showing you here is what we refer to as our dashboard view. This is just a quick summary of the output of that phenotypic screening, showing all the in vivo model tests that we ran it through. And activity is shown in the case of green, means it's statistically significant in a therapeutic direction, and yellow means that it's trending but not significant in a therapeutic direction. But just to give you the gist of what you're looking at, by the way, we didn't have gastric ulcer on this panel. We saw a constellation of activity all around metabolic disease. So just to rhyme it off quickly, we saw activity in the oral glucose tolerance test, decreased weight gain when animals were exposed to high-fat diet, improved leptin levels related to that weight gain effect, and increased resistance to cold sensitivity. So that, by the way, I told you Pfizer was first was working with this drug, but they were working in an era when that really preceded target-based medicinal chemistry as we know it today. So in other words, they didn't know what the molecular target was. And in this day and age, we knew that that was really an essential element, although it's not a regulatory requirement, is nonetheless from sort of partnering and investment point of view, we considered it a requirement. So there's a lot of work that we did following that I'm just kind of summarizing here by way of showing the headlines of the papers that we've published around that. But the gist of that was through partly good fortune, but a lot of screening of panels, screening and so on. We identified that the target of this drug was LRIG kinase, and it was an activator of LRIG kinase that was reasonably potent, that is at a double-digit nanomolar EC50. And we really proved it unequivocally in the sense that when we look at LRIG knockout mice, for example, we lost all the glycemic control pharmacology that we'd identified. So I think in some regards, that's where we first, I would say we first sort of connected and made LRIG kinase a therapeutic drug target, was at that point. And so what I want to do now is to sort of quickly walk you through a series of chapters, if you will, on different therapeutic indications. But the first chapter I'm going to walk you through quickly is it's the relationship of LRIG kinase and MLR1023 to type 2 diabetes. Obviously, this was something we discovered early on. I'll just give you a quick snapshot of some of the data. In the field of diabetes, one of the requirements for any product in this day and age is to work well with metformin, because metformin is and will be for a number of years to come a first-line therapy. So you've got to be able to pair well with metformin. Here we're comparing the activity to metformin. It's comparable glucose lowering to metformin in an oral glucose tolerance test, but you see when they're paired, we got an additive effect. So it's clearly a complementary mechanism. But more, really the exciting part is that we showed that this was an insulin sensitizer, and that's illustrated here in data from a hyperinsulinic euglycemic clamp. So the way this works is animals are infused with a constant rate of insulin, which of course has an effect of lowering blood glucose. And then at the same time, we infuse with a variable rate of glucose, that rate which is required to attain some preset level of glucose, in this case, say 100 mg per deciliter. If they don't, if they're not insulin sensitive, it's relatively low level of glucose that's required. But if they are have increased insulin sensitivity, those glucose infusion rates are higher, which you get with a true insulin sensitizer, such as a glitazone, if for those who are familiar with that class of drug, rosiglitazone, for example. So MLR1023, like rosiglitazone, is effective in this. There's no other class of drugs other than true insulin sensitizers that are active in this model. So for example, the SGLT2s, the DPP4s, the GLP-1s are not active in this, but MLR1023 was. And the reason that's important is because the glitazones are a very important class of drug that is, for all intents and purposes, disappeared because of other sort of class liabilities that they have. All the glitazones were PPAR activators, and they come from a class, structural class, referred to as TZDs, or thiazolidine diones. So what we subsequently found is that MLR1023 is not a PPAR activator, has none of the sort of characteristics, does not activate in any assay for PPAR. And you can see from structure here, I'm showing the structure of MLR1023. Medicinal chemists would recognize that there's not a structural similarity to the TZDs. So that was very encouraging. I mean, here I'm just showing a little bit more that illustrates the point of its insulin dependence in another way. Here we're looking at a type 1 model. So this is a STZ rat, which means that the animals are treated with a toxin, streptozin, that ablates the pancreatic beta cells, so that in this, and you see, first you see got elevated glucose levels at the baseline here. When we treat with MLR1023, there's no drug effect in those animals. If we treat with insulin, shown in blue, you can see lowering and then regain of blood glucose. But when we treat with the same dose of insulin and MLR 1023, now you see drug activity that is represented by a potentiation and prolongation of that insulin effect, possibly meaning it might have benefit in type 1 setting as well. But I think it illustrates the insulin dependence on for this mechanism. We've, as Dennis alluded to, we have taken this to the clinic and successfully proven in our phase 2 studies that we get the same glycemic control effects in the clinic. And this was a small POC study, 23 subjects per arm. But here we're comparing the results historically, not head-to-head, with what was then a much newer class of drugs, a DPP-4 in this case, a Januvia or Sitagliptin. So you can see we have sort of commercially meaningful blood glucose lowering in this 4-week study. So I want to wrap this portion of this chapter up by just making the statement that I think the story also illustrates that it is necessary but not sufficient to prove that the drug is safe and effective to ultimately be successful in taking a drug to market. And what I'm referring to is that there are many more factors other than just the proving it's safe and effective. And in our case, with a drug that might launch in sort of a 2025, 2026 timeframe, we have exclusivity through the patents that we've attained that would take it out to 2031. In this sort of setting, and I should point out too that we've done extensive commercial assessment working with our potential licensing partners. These would be, of course, sort of multinational pharma companies that are active in this diabetes space. So we've spent a lot of time doing commercial assessment around this. That with that period of exclusivity, and by the way, for example, hundreds of physicians active in the area, payers, and so on were involved in sort of this commercial assessment exercise, and where we came up with projections, mid-range projections of peak sales estimates from that exercise of well in excess of $1 billion. Even at that, that does not sort of meet the hurdle rate required. As you can well imagine, peak sales of $1 billion or more are not attained overnight. It takes years, maybe 5 or 6 years to achieve that. And with the costs, which in this case, in the case of type 2 diabetes, being hundreds of millions of dollars in commercial trials still between now and a market launch, those factors do not sort of line up to make for a positive net present value when you look at the costs and the benefits at the end of that. So this is sort of what where we are with the type 2 diabetes story, but and that's kind of the lessons learned. So now I'd like to shift gears and talk about sort of chapter 2, Lin kinase in obesity. I told you that we discovered effects on weight gain, but there's really some fascinating science that has evolved since we first made those observations early on, and that I'll walk you through very quickly. So this is just showing some of the early data. For example, animals turned onto a high-fat diet, and you can see the significant lack of weight gain in the highest dose group here, which, by the way, is not explained through altered food intake. Food intake is normal in those animals. This is just some additional data. In this case, we're looking at a combination with rosiglitazone. As I mentioned, there are some class effects with the glitazones, one of which is cardiovascular rich effects, which is associated with weight gain. It's well known that the glitazones do cause weight gain, and here you see it in the red line. So these are the rosiglitazone only treated animals. But when we do the combination, the 1023 completely blocks the weight gain that would otherwise be seen with those. So it was years later when we were doing our clinical studies, we became involved with a site, the Pennington Biomedical Research Institute, which is wholly dedicated to metabolic disease, diabetes, and obesity, and working with an investigator there, Frank Greenway and Ann Coulter, who were very fascinated by some of the preclinical data that we had, including the weight gain. And Frank formed the hypothesis that this really had all the hallmarks of something that may be causing a phenomenon referred to as browning of white adipose tissue. And for those who aren't familiar with what that means, there are two types of adipose tissue that all mammals have. White adipose tissue is the fat tissue you're all familiar with, and then brown adipose tissue you might be less familiar with. That's something we as humans are born with, but becomes vestigial within a few months after we're born. But otherwise, in all or maybe at least most other mammalian species, it remains a very functional and prevalent form of adipose tissue. And its role is really thermogenesis. And that is to generate heat, which is why our dogs and other animals can survive the cold as well as they do. And the way they accomplish this is through mitochondrial uncoupling. That is, the mitochondria undergo respiratory processes that they do, but without ATP. So it's essentially spinning their respiratory wheels and working with. There's also signature gene expression that's associated with around involved in fat oxidation, all its generated that effect. Maybe one of the real hallmark markers of that is uncoupling protein 1 or UCP1. And one of the things that they do by Pennington quite frequently is to work with primary human adipocytes. And when they took human primary adipocytes and treat them with MLR1023, they saw gene expression induction that was indicative of browning, and particularly very marked increases in UCP1. When we went back and looked at some of tissues from studies that we'd done previously on chronically treated animals, we also noted in animals, in mice, marked increases in UCP1 in those animals as well. What we also found, and this goes back to the clinical study that I've already referred to you, that phase 2 study, although it wasn't designed as an adiposity study, nonetheless, we did see statistically significant reduction in weight in that 4-week period. So that was a 4-week treatment, and here's the most active group at the end of treatment and at the 1 day or 1 week follow-up after treatment. And that's highlighted even further here in this dashboard view of the study. So here we're looking at the various treatment groups and then lipid levels as well as weight. And green in this case means statistically significant in a therapeutic direction. Blue is in favor of drug or in a therapeutic direction, but not statistically significant, and red is no effect. So what we see across the board, except for HDL, is an improvement in lipids, that is lowering LDL, cholesterol, triglycerides, and free fatty acids, even when it wasn't significant. And as I say, in our most active group, statistically significant weight reduction. So where does that bring us? And essentially, we feel it's a similar story to what I just told you with type 2 diabetes, that our intellectual property, our exclusivity window is really getting long in the tooth on one hand. It would take us out to at least 2031 or 2031 specifically. But we couple that with knowledge that clinical development for obesity drugs has proven, and we see this from others that have gone before us, to be very challenging. So we put those two factors together and say this would be sort of a tough row to hoe, so we're not currently pursuing that. Chapter 3 that I want to tell you about then is Lin kinase in NASH. And for those who aren't familiar, NASH refers to non-alcoholic steatohepatitis, which is a disease of fat accumulation in the liver that leads to, in its more advanced states, ultimately liver fibrosis, cirrhosis, and has become a leading cause of liver transplantation now in this country. Currently, there are no therapies, approved therapies for NASH. It's to the extent you haven't, some of you may not have heard of it, it's because 10 years ago it wasn't as, wasn't as much awareness, but through increased imaging modalities, it's become, the awareness has increased tremendously. And is making it sort of much more an area of pursuit for pharma companies. And by some projections, because the number of people, maybe something on the order of 20 million people in this country that have some form of NASH, some people have projected market opportunities on the order of $30 billion. So, like I told you that there was an investigator through sort of an unbiased approach, who when I was telling you about Mueller discovering the relationship of Lin to the insulin signaling pathway, equally, there's a story about an investigator studying liver regeneration. And so this lab was looking at factors responsible for driving hepatocytes down a regenerative pathway versus an apoptotic pathway, and that apoptotic versus regenerative balance that exists. Their focus around apoptosis led them to further focus within mitochondria and factors involved in that. And long story short, they discovered a key factor responsible for driving cells toward that regenerative pathway was Lin kinase. So given that NASH is sort of a juxtaposition between metabolic disease, because it's insulin resistance and fat accumulation are a key component of it, but it's an intersection with liver degeneration and liver fibrosis. We found that sort of an intriguing combination within Lin that sort of warranted further investigation, which we've undertaken. And so again, a lot of work was done here. We've run this in multiple animal models, and NASH models, by the way, are very long duration, typically on the order of about eight month duration models, which for preclinical models is certainly long. That I'm summarizing here. So in the first model we ran, this is a form of modified high-fat diet that the animals are turned on to. At the 8-month mark, which is as long as we were willing to go, they developed key hallmark features of NASH, and the one I'm most focusing on is this non-alcoholic fatty liver disease activity score noted here. But you have to take them out another 3 months or so to get some of the more advanced features of the disease, including liver fibrosis, which we hadn't done. So we weren't really able to look at liver fibrosis in this first model, but nonetheless, we were hitting on all the other kind of key features of NASH, which is sort of, for those who are familiar, it's a multipartite disease. The key features, of course, being that histology, the liver histology component, but things such as insulin resistance and adiposity are really core factors in the disease as well. And so we were what was most notable is that we were hitting on those multiple features. So something we did, because we were missing liver fibrosis, we looked at liver fibrosis unto itself, not in a true NASH setting, but in this carbon tetrachloride liver fibrosis study, where you're looking at a form of liver fibrosis independent of metabolic effects that are a component of NASH, which ultimately turned out to be quite interesting, because as you can see, I mean, we did find activity with MLR 1023, which, by the way, I it wasn't obvious to people. Green means statistically significant. Yellow is trending but not significant. And so we saw this nice dose dependent effect of therapeutic effect of in liver fibrosis. We didn't see any effect on liver enzymes, but we did see it on fibrosis per se. And so what's interesting about that is then subsequently we've looked at another NASH model, which is somewhat like the first model we did, but with a very low levels administration of carbon tetrachloride, small enough that they would never affect the liver under themselves in a normal setting, but in the context of this modified high fat diet, they sort of push the fibrotic feature along a little bit. And so there again, we saw the dose-dependent antifibrotic effect, as well as things we've seen before. But the point I was starting to make here is that in this carbon tetrachloride model, we're showing that this is a direct antifibrotic effect. It's not antifibrosis in this case, secondary to our having effects on improvements on metabolism. And here I'll just quickly, sort of show some of the data. Here's some of the histological features, which can be, I'll just tell you, I mean, it's quite obvious even to the untrained observer when you look at these livers. And here's the antifibrotic effect. This one comes from the CCL4 model that we ran. I would sort of score this as it is a sort of an or a rather mild antifibrotic effect histologically speaking, but nonetheless is picked up by trained observers and is statistically significant when we measure it using markers for fibrosis, such as hydroxyproline, where we can quantitate it. So where does that bring us for NASH? We've sort of solved our IP and exclusivity issue with fresh intellectual property on for using 1023 in the field of NASH, but somewhat like with diabetes, we've watched the field sort of evolve beneath our feet in the timelines that we've carried out this research. So all along, it was true, and it was the case that that to take a product to market in NASH requires very long treatment times, and these are dictated by the FDA. So we're looking at about 72-week treatment times with well over 1,000 subjects for the registration studies. And all of those patients, again, for the registration studies, have involved 2 liver biopsies, a liver biopsy at baseline and at end of study. So you got to get well over 1,000 subjects who will volunteer for 2 liver biopsies. So it's made NASH arguably one of the most difficult areas to develop in. And now what's happened more recently is the first 3 companies to brave that pathway have failed. So it's sort of changed the tenor, changed the level of excitement in this field, and that affects us. So we maintain our patents in NASH, but it certainly sort of puts it, let's just say, to put it mildly, puts a damper on things. So now I want to turn to, I guess we're up to chapter 4, and talk about LIN kinase in type 1 diabetes. And there's really some things, as I'll show you, that are taking place in type 1 diabetes that from a therapeutic point of view set it apart from type 2 diabetes. And as I say, I'll get into this in more detail. But early on, we knew that there was something going on in beta cells. Here I'm showing you data from DB mice. And so DB mice recapitulate type 2 diabetes in the sense that it's a progressive form of insulin resistance, and there's a compensatory response in the pancreas, whereas insulin resistance is set up, more and more insulin is made, and eventually the beta cells in the pancreas become exhausted and degenerate, as you see from these ghosts here in the vehicle treated DBDB animals. The 1023 treated animals, we noted early on, were preserved. And you can see that too by preserved insulin levels compared to the vehicle treated animals. The problem at this point was we didn't know whether this was a primary effect or secondary effect. Was it having direct cell survival activity on beta cells, or was it secondary to the fact that we're relieving the beta cells because we've restored insulin sensitivity? That part could not be determined initially. More recently, we've done these studies with DBDB mice again, wherein here you see the decreased fasting plasma glucose levels on the 1023 animals versus the DBDB vehicle treated animals, and here compared to lean animals. But when we discontinue treatment, and what I need to tell you too is that 1023 is a drug with quite a very short half-life, a plasma half-life of only about 2 hours. So within 24 hours, all the drug is gone, and yet for well over 2 weeks, we see a sustained separation of the glucose. So there's something transformative that has taken place that is able to keep these two groups separated. And we think that this was evidence that there was a transformative change in the pancreas. But what was really exciting, and this is I think the most exciting data that we could potentially talk about, but unfortunately, I'm somewhat restricted. This again is data I'm referring to from a third-party collaborator who he's a good guy, but he's not as comfortable sharing the data as I presented to you from the Pennington Biomedical Institute. So I'm not able to sort of show you all the exciting data, but I can talk through the headlines. And so here I'm referring to an investigator named John Buteau from the University of Alberta, which is a World Center of Excellence in beta cell biology. He's dedicated his entire career of, I don't know, something maybe 20 years or so, and to beta cell studies. And at least for the last half of his career had been studying factors that promote survival of beta cells, for example, in the condition of glucolipotoxicity as beta cells encounter in a glycemic condition. And again, in an unbiased way, as I told you about with Mueller and others, he used proteomic and genomic approaches to narrow down on the factors that were responsible for conveying and making the beta cells resistant to apoptosis. And his studies led him to Sark family kinases, and eventually he's narrowed it down to specifically Lin kinase. And he at that point, he was unaware of our work. We were unaware of his work. And so he then contacted us because of work that we published related to diabetes and Lin activation. We're the only company that has a Lin kinase activator. And so he found us, and so our two worlds coincidentally came together that way. But since that time, he's done a lot of studies in multiple complementary systems in an immortalized beta cell line in rodents. But as being at a center of excellence as he has access to primary human beta cells. And so in all these systems, he's done gain of function, loss of function with Lin kinase, and then used 1023 in all these. As I say, it's a very large compendium of work, which I hope will finally be published maybe within the next 6 months or so. So I would encourage you to look for it, because it really is some beautiful work. But I'll just give you the most exciting headline feature here, which is he can take a cell line that was previously thought to be proliferation incompetent, primary human beta cells in adults, take them from cadavers and treat them with 1023 and get those cells to divide. So that is something that we're now actively pursuing with him. And I just want to tell you a little bit about that. So C-peptide is a fragment that's released from the production of new insulin. So it's a biomarker of new insulin. And the reason that's important is because if we're looking in, well, let me just tell you this next thing, that about 20 to 30% of type 1 diabetes patients have detectable levels of C-peptide. And that's an important biomarker because remember, we're providing them with exogenous insulin. So C-peptide provides a marker to show us that there's endogenous new insulin being produced. And so that's the function in this context of C-peptide. So what does that mean? Why did these, by the way, are type 1 patients that have had the disease for many years, a decade or more. They're stable, and yet they have remnant beta cells that are producing insulin. We don't know, the experts in the field don't know whether that they represent immune privileged cells, or somehow that the autoimmune assault that that wiped out the beta cell population largely ceased before it completely wiped out all beta cells. It's not known, but our intent is to take subjects of that type, treat them for some period of time. Our protocol specifies for 4 months, and see if we can move the needle to increase levels of C-peptide. If you can, of course, the next question is, can you continue to do it over a period of time? With the ultimate goal being that you may be able to reduce insulin requirement in one sort of minimum, or even as sort of an ultimate goal, perhaps cure type 1 diabetes in these individuals. And I should point out that Buteau has been able to cure a recapitulated form of type 1 diabetes in mice with 1023. So it's not out of the picture. So this is an active area that we're pursuing. I now want to turn to my last chapter quickly and talk about a very different area of biology, pulmonary inflammation. And so again, as I've told you before, Lin has popped up in the literature in other ways. It turns out that in sepsis and the cytokine storm associated with sepsis, there are cytokines that are linked to certain kinases in pulmonary epithelium that lead to the phosphorylation of cadherins in a pulmonary epithelium that opens pores and causes pulmonary leakiness and edema in the case of sepsis. And as I say, the pathway has been worked out, but more recently, it was determined that an opposing factor to that pathway was Lin kinase, and Lin kinase, in short, is a very important factor in sort of opposing that leakiness that ensues in certain cytokine storm conditions. Obviously, now we'd been aware of that for some years, but obviously sort of last March and April, you can imagine it didn't take a tremendous leap of faith to realize that this is something that could be pursued in the context of COVID-19. So the first thing we did was to look in these acute models of sepsis. This is an LPS challenge. In this context, we, in our first study, to give it the best shot, we treated animals subchronically for 7 days, including 1 hour before we presented the LPS challenge. And the vehicle-treated animals that you can compare to are these this group in black. And then what we've looked at is edema by a couple different methods, either Evans blue content, which is a high molecular weight dye you introduce intravenously and leaks into the lung if the lungs are leaking, or we can do a wet weight, dry weight. By both conditions, we saw significant reduction in edema in those animals. In the next study, what we wanted to do is to figure out how to investigate better how this might be treated in a clinical context. In other words, is it therapeutic? Is it prophylactic? And so what we did is just took that dosing paradigm that I just described to you and broke it into two. This group here marked as 7X, it was the same dosing regimen I just described to you, 7 days and then 1 hour before. This one, the 6X, we treated subchronically, that is the 6 days before, but then 24 hours prior to LPS challenge, we did not treat further with 1023. And then a day later did the LPS challenge. And this group is just doing that 1 hour prior to LPS challenge component of the 7X. And so what we find is that all the benefit, or essentially all the benefit, was conferred well prior to the challenge. In other words, we interpret this that we provided a prophylactic benefit to the cytokine storm and the pulmonary leakiness that ensues. We next took this to a subchronic model. We couldn't work with COVID-19 virus, but we were able to do this with influenza, which also has a similar sort of phenomenon, a cytokine storm. And in that model, we've looked at survival, have a significant survival benefit and recovery shown here, looking at body weight. And that translates again to an improved, in this case, wet weight, dry weight, reduced pulmonary leakiness when we treated animals. And by the way, this is starting at the beginning of their influenza infection. I just point out that in both those models, here looking at the LPS model, but in both those models, we see no evidence for immune alteration. So in the case of the LPS study, we look at key cytokines that are not altered in any way. And in the case of the influenza study, we look at B cell function, at total IgM, total IgG, and the flu-specific antibody, and see no evidence for altered immune therapy. So it's very consistent with this is a pulmonary barrier therapy. And so where we are with that, in less than a year since this work began, we now, as of February 1st, just filed our IND. This is, by the way, in part with support from state of Pennsylvania. And as of today, as we discuss this, we were just cleared today, so that IND is now active. So going forward, we're very interactive with the NIA, which the money's not in the bank, but we're hoping, but we'll see, looking to get support for this work. That's the National Institute of Aging, as I say, with some active, an active program that we've been discussing with them. But the real challenge, of course, is once again the world evolves in the timelines that this work goes on. And so the nature of the study that we're looking to do, because it is a prophylactic, we're looking to treat patients as early as we can get them before they develop pulmonary complications. And then our outcome measures are to look at the percentage of patients that develop pulmonary complications. In order to do that, we've got very specific recruitment criteria, so we don't have very large patient numbers. So we're taking subjects that are 7 years of older with at least one risk comorbidity, such as diabetes, obesity, chronic kidney disease, or cardiovascular disease. So, so trying to recruit them in the evolving environment, you can see, I mean, is the challenge we currently face. But if we are able to pull this off, the optionality, that is the other disease states we would then go into and have confidence in having effect would be large. So that's sort of the upside that still lays before us. So those are my multiple chapters. Just conclude by reiterating my message that biology is complex, that I tried to make an argument for why there's much more beneficial biology in the targets that we're working with, and I think the field appreciates that. I refer to as pharmacological pleiotropy. I've illustrated that with numerous examples with Lin kinase and 1023, each one having a scientific underpinning, animal model data in each and every one of those sort of chapters. And 2 of those were clinically validated in the case of diabetes and obesity. But of course, the other message too, as you see, that even doing the hard work of showing that a drug is safe and effective in the clinic is not, that there's much more to ultimately having successful drugs. So I'll leave it there and take any questions.
Thank you, Andrew. Excellent.
Yeah, I
We
just wanted
do have
to remind people to make sure to select the drop down to all panelists, and Ron will read the questions. Thank you.
Great, thank you. We have a couple of questions, and I'll start with, were all the different therapeutic indications, and I guess it's referring directly to MLR 1023, were all of the different therapeutic indications expected based on your known biology or tissue distribution for that target?
No. Again, I mean, expected at some point in time as people turn up evidence. But if you go back to, I mean, the short answer is no. There's been serendipity all along, and I think I'd leave it at that. I could expound, but the short answer is no.
The next question goes up, I guess, to a little higher level, like it would be addressing the early days of screening and for repositioning. Do groups or companies that focus on approved drug repositioning normally focus on specific disease areas, or do they screen broad targets to find opportunities for further investigation?
Right. So I think the answer is both. There are companies with a focus, and particularly you see this a lot in the not-for-profit areas, such as Tuberculosis Foundation, that have a specific disease focus, and they try to address that disease state in most cost-effective way using a repositioning strategy. So there's lots of examples of that. And then you have some examples, companies such as us, the company Recursion, that are start with a drug and try to find so being as opportunistic as they can, but are disease agnostic, I would say. So there's good examples of both.
So I guess a follow-up question on that is, let's take a company like yourself that is looking at a drug. Are there ways to increase the screening using something that would be equated to like a blast search in genetics to expand the potential targets?
Well, I mean, you're asking me, and I have a very specific bias, but a basis for that bias. And if you had another hour, I'd give another lecture on our belief on phenotypic screening. So yeah, so my short answer to that would be I believe that phenotypic screening, because it is unbiased, because it is not limited by what I described at the beginning of that that proportion of how much do we know and how much don't we know, and what are the effects of all that. Phenotypic screening is not limited by that problem. So my answer to that is phenotypic screening.
Okay. At this time, I don't have any other questions that have been posed to the panelists. I'll turn over to Rodney for closing.
Yes, thank you. Thank you, Andrew. We appreciate the excellent talk here today. And thank you very much. I invite everyone to join us 1 month from today for a talk by on dual presentations on high throughput cellular thermo shift by a group at GSK and NIH NCATS. Thank you very much and good afternoon.